going to keep this as an informal as I can presentation, because you have now all had probably as much in the way of virus information as you can stomach for 1 year, and you're preparing for your exam. And probably something that you hear over the next hour or so will be on that exam, but I'm not sure about that. Vince, what do you think? Will be, okay, so you have to pay attention. I would have given you the afternoon off otherwise. Okay, so I think one of the themes that has run through this course is the relationship between the environment and virus infections themselves. How do we catch these things anyway? How do they get into us? How do we transmit them from person to person, for instance? Or do we? And if we do, then what are the mechanisms involved? Influenza is pretty easy to understand because of droplet infection. Everybody got that one. Wash your hands, cough, covering your mouth. Those are all some of the easy public health measures to keep the spread down, but not completely out. But then there are some other more complicated ways of spreading infections. We can spread them by oral, right? We can eat something and catch it. Know any viruses that you catch by eating? Polio, roto, sure. Another way to catch a viral infection is through sexual contact. So we know about HIV. We know about papillomaviruses. And then there's the vector-borne way of catching them, right? So you can have them transmitted to us by something that we can't control, or at least not easily. So mosquitoes are one of those things. Ticks, fleas, lice. Bed bugs? Know of any diseases spread by bed bugs? I don't either. In fact, there aren't any. But they've tried to prove it again and again. They had a main candidate for wanting to know whether or not hepatitis virus was spread by bed bug bites. Anybody here know what a bed bug looks like? No. You live in New York. No, there are lots of bed bugs in New York. It's made a big comeback, in fact. Over where we work in the Uptown campus, bed bug heaven. It's like endemic. That wasn't always the case, and no one actually has a clear understanding of why that is. Why are there so many bed bugs now as opposed to, let's say, 10 years ago? You can see signs in all the drugstores now. They have medications for bed bugs and repellents and all that stuff. It's kind of a mythology that's grown up for bed bugs. There was a guy here that was actually a graduate of our medical school who went on to win a Nobel Prize, which is pretty good stuff. His name is Baruch Blumberg. And he spent, I would say, 20 years of his life trying to show that bed bugs transmitted something. He spent most of his time in West Africa because he was an expert on hepatitis virus. And that's where he won his Nobel Prize, for proving that hepatitis is caused by a virus. Have you studied hepatitis virus? And you know about this guy, Baruch Blumberg? Ah, why didn't you tell him? It's one of our local heroes. No. I'm going to talk about something else. I actually met him. He still is a wonderful person. He's now in his 80s, and got his ideas from travel. So when he was a medical student, he got to travel to South America. And while he was there, he noticed that a lot of people had hepatitis, and he isolated a protein from their blood, which matched with another protein that someone had isolated from the island of Australia. And he called it the Australia antigen because it matched identically with that. And it turned out to be the surface coat protein from hepatitis B virus. And because he put two and two together, he was able to discern that this protein was associated with the virus infection, and the virus infection was associated with liver cancer. That's interesting. So hepatitis is a virus infection that in 23 to 25% of Asian males without vaccines would cause liver cancer. So with a vaccine, you can prevent cancer by preventing hepatitis. And that was worthy of a Nobel Prize, too. but someone else got it for that. At any rate, so what I want to tell you about is another virus infection. There are so many. In fact, there are too many. But there are some worth noting, and that's this one. At least I think so. Why? Because in your lifetime, this virus infection, A, wasn't here, and then B, was here. So this is the story of how the West Nile virus came to the New World. Well, that means that it had to come from the Old World, right? It's the same age. I don't know why we call one, I guess because it was discovered later in life, so with regards to the human condition. So the general topic is medical ecology, and it's the study of the environment as it relates to an issue in medicine, in this case, how viruses are transmitted from one person to the next. And since that doesn't show up very well, I'll read it for you. It says West Nile virus in North America. And again, it doesn't show up very well, so that's a mosquito biting somebody over here. This is an electron micrograph of the virus particles. And this is a sketch of its main host species. And that's not us at all, is it? So do you know of any other viruses that we can catch which we're not the main host for? Anybody? Oh, come on, you know the answer to these questions. What about rabies? Rabies, right? Who's the host for that? Sorry? No. Does anybody know who the main host for rabies virus is? Keep your hand down in the back. He doesn't have to take the exam. He's exempt. Sorry, who said bats? Is it baseball bats or just regular bats? Just bats. Flying bats. Flying, the kind that, okay, as opposed to the kind that crawl. Okay, so all bats fly, mostly, or they glide. And did you know that 20% of all the mammal species on Earth are bats? That's a lot. 20% of all the mammals that are species on this planet are bats of various kinds. And many of them, at least in this hemisphere, carry the rabies virus. When I say carry, that means that they don't get sick, right? That means that they're well adapted to their virus. That means their immune system or something keeps them from becoming sick. This is supposed to be a crow. Let's imagine this is the crow. And let's imagine this is the virus, which we don't have to because it's there. And there is a mosquito here that prefers to feed on birds rather than on people. I'm giving a little bit of the story away, but that's the story that I'd like to tell you. Because if this is the preferred host, and if this is the virus that infects this animal, it also infects an invertebrate. So right away it tells you that it's got a very large host range. Very large. In fact, I believe that this virus has the largest host range of any infectious agent ever. Do you know of any exceptions to that, Vince? Or Saul? You know of a virus that has a larger host range than West Nile? It infects more insect species and more cold-blooded and warm-blooded vertebrates than virtually any other infectious agent on the planet. So how come you haven't heard about this before? I should have been your first lecturer, not your last. No, that's not true. So I'm a parasitologist by training. I'm not a virologist. And anybody listening to TWIV or TWIP would know that clearly by just listening to my stupid questions in virology. Because I wasn't trained as a virologist. I was trained as a parasitologist. So what's the difference? The difference is that we deal with much bigger organisms that we can see. I don't trust an organism I can't see. And I can't see these viruses, so I don't even know they're there. It's like the emperor's new infection. That's not fair. So I like a worm. Give me a worm any day. A nice big old fat juicy worm. And that's what I did 27 years of research on in my laboratory. So when the NIH finally told me that I had done enough research in my laboratory, which was a very bad piece of news to receive, of course, because I wanted to do 28 years worth of research, and they only let me do 27 years worth. It forced me to do something else. So I said, well, what else can you do? That was a rhetorical question, of course, because I didn't have an answer. The university, this is a great place, right? I mean, I know you pay a lot of money to come here. And they'll ask you for more when you leave. So be aware of the fact that this doesn't end here. In fact, this is a pittance compared to what they're going to expect from you when you become rich and famous. But if you become rich, they don't care if you're famous. They just want your wallet. And this is recorded, I hope. Okay, so point is that this university is great for many reasons. And I think it's a great place to be at. Because what they told me was, well, we appreciate all the nice hard work you've done for us and all the grant monies that you've got for us and all the fame that you've brought this place in terms of your own career. And now you have a chance to do something else. So what else would you like to do? How often do you get that chance? Never. Never. Trust me, never. But if you are given that chance, jump on it. Just jump on it. So I did. I actually, I said, what have I been doing that I enjoy the most that doesn't involve laboratory work? Because that's what they said stop doing. Not because I was doing it poorly, it's just because they wanted to give the money to somebody else to do it on something different. All right, so I love teaching. And I also love thinking about the environment and how we interface with it. I've spent my whole life contemplating that, and it's because I love nature, and I think that's why you're in this course, because you have a love of nature. You want to know more about it. You want to know what makes us sick. You want to know how to avoid becoming ill. Perhaps some of you will go on to become doctors, engineers, lawyers, but you'll always have this interest in nature because viruses are an integral part of nature. And this is not wasted education at this point. You're learning stuff it'll stay with you for the rest of your life. So this university said, we can afford to have you change fields if that's what you want to do. So the value of a good general education is to allow you to do that. So that's another thing to keep in mind here. And that's why this place is also great, because it's a big menu to choose from. So I began to rethink how to evolve into something more generalist, and how to pay more attention to the bigger picture rather than the smaller picture that I was working on for many years. And so one day, in July, a medical student came to me and said that she had 2 months of time free before she started her clinical rotations as a first-year resident in infectious diseases. Please turn off your cell phones. But that wasn't a cell phone, right? It was a computer. Please turn on your computers. Your shoes. All right. No. So I said, her name was Barbara Nichols, and I said, Barbara, there's been some strangeness about the recent past. This was late July, I guess it was early August, actually. And there have been some illnesses in New York City that were largely unexplained, thought to be caused by a vector-borne virus infection. And they blamed it on St. Louis encephalitis, because we have St. Louis encephalitis virus in this country. It's carried by mosquitoes. Sometimes people catch it, and they get sick. And if some of them get sick, occasionally somebody dies. And that's what had happened recently. And the samples that the Public Health Service sent for analysis came back from the Centers for Disease Control positive for the St. Louis encephalitis virus. Case closed, right? No, not if your house. It's not closed if your house. What would House have done? I know what House would have done. He would have taken all the residents and lined them up, abused them verbally, and then accused them of being all stupid, except the one that could solve the problem, who could get the prize behind this door, which was a half-drunk bottle of single malt scotch, because he drank the other half. So someone starts to speculate as to what might be wrong with this picture. What was wrong with the picture was these little critters right here were dying by the dozens throughout the metropolitan area of New York City. Now, that doesn't sound so strange, does it? Come on, does it? Dead birds, hey, you see them all the time. Dead pigeons, dead sparrows, dead starlings, dead crows. Dead crows, you ever see a dead crow? Dead crows. A little bit different. You know what you call a group of crows? A bunch of crows? It's a murder of crows. That's right. It's a murder of crows. So somebody had murdered the crows. Now, what are you laughing at? This is not a funny subject. This gets much more serious in a minute. Because House sends his crack staff out into the parks to pick up dead birds and bring them back to look at underneath their super electron microscope. Does that really happen? That's not actually what happened. The crows started to die all right, but they were dying mostly up around the Bronx Zoo. Bizarre. Outside the Bronx Zoo, not inside, outside. At the same time, of course, inside the Bronx Zoo, there was this fabulous exhibit of gorillas that had just been shipped over from Central Africa. I don't know if you recall that or not. It was a long time ago, in 1999, in the summer. And an entire troop of gorillas were essentially on vacation in New York, spending their time at the zoo during the day, and then at night they'd take in a play, go down to the village, grab a pizza. And I went to that exhibit in September, and those gorillas were standing behind the plexiglass, and they put their eyes right up against the glass to look at you looking at them. And they would look at you and say, "Where the hell are you from? Who's your people?" And you'd look at them and say, "You know, I resemble that remark. We have a lot in common here, you and I." And there were 19 of these things that you could look at, and it was quite amazing. It attracted so many people that the lines were 3 hours long. Because I told you this for a reason. 3 hours long you had to wait online. And it was hotter than it was now. And people were wearing shorts because it was the summer. And they were busy doing two things. One is trying to keep cool. This was the Congo exhibit, and there was a mist of water that was sprayed out over the trellises so that you could stay a little bit cool as you were waiting online. But the people on the outside of that line were hot. So they were doing a lot of this. What do you think they were doing? Sure. And where do you think the mosquitoes were coming from? This is even more interesting. Right over there. Where? Just there. Where was there? In the Egyptian goose pond. If you ever want to see a body of dirty water, go to the Egyptian goose pond in the Bronx Zoo. It's a cesspool. It's the dirtiest, ugliest water you can possibly imagine. Does that have any bearing on what I'm about to tell you? Damn right it does. So here we are, standing online, waiting to see these lowland gorillas, not the upland silverback jobs that you and I are used to seeing and beating their chests. And these are the lowland, gentle types. Come on, they're friendly. And we're being bitten alive by mosquitoes that are breeding in the filthiest water you can possibly imagine. At the same time that that's going on and the crows are dying outside, something else is going on too. We had 5 encephalitis cases, 2 deaths. A positive serology for St. Louis encephalitis. And inside the zoo, other birds were dying. So now you're Tracy McNamara. You're looking at me with a sconce here. So I'll ask you a question. You hate questions, but this is a prelude to the exam. So Tracy McNamara, who was one of the world's leading authorities on bird diseases and the assigned pathologist at the Bronx Botanical Gardens and Bronx Zoological Gardens, was assigned the task of autopsying the dying birds inside the zoo. Okay? Take you back to House. Line the birds up and tell me whose birds were dying. I'll give you some birds. South American flamingo. Himalayan duck. A Cory bustard. Nope. A Cory bustard lived. A Cory bustard was sitting in the cage between the Himalayan duck and the South American flamingo. The South American flamingo died. The Himalayan duck died. Cory bustard? Could care less. Where are Cory bustards found? You know that bird at all? Anybody here ever been to anywhere in Africa? But okay, so someday you will. In Africa, the Cory bustard rules. It's a snake hunter. And it's a very unusual bird. So if you ever get to the Bronx Botanical Gardens in the Bronx Zoo, check out the Cory bustard that didn't die from this whatever was going on. But the other two birds did die, and there were other birds that died too. And virtually every bird that died was not from Africa. And virtually every bird that did die, no, the other way. I misspoke. Every bird that lived was from Africa, and every bird that died was not from Africa. And meanwhile, outside, the crows are dying. And meanwhile, inside the zoo, the people are going like this. And we've had 3 deaths, and they blamed it on St. Louis encephalitis, which was an endemic infection in the United States. And oh, there's one other fact that you have to know about, and that is that no birds within the continental United States die from St. Louis encephalitis. Although they catch it, and they transmit it from bird to bird, they don't die from it. No birds die from it here. But I bet you if St. Louis encephalitis was introduced to the Bronx Zoo, all of the exotic birds would die from it. Does that sound like a familiar pattern of other viral infections, like, for instance, smallpox, or maybe even influenza, which, if you're used to it and your population has genes for resistance, you don't die, but if your population has no genes and they've never seen the virus before, you all die? Like, for instance, in the New World, when the Spanish came, and there were 50 million people living here. And then 250 years later, when they left, there were 4 million people living here. They didn't have enough bullets to kill that many people. But their infections were their bullets. That's what happened. Okay, so that's a good case of a population controlled by a parasite. And then what happened was, of course, the Spanish left their genes here in the form of little children from the rape victims. They grew up to repopulate, and those diseases would never affect that population again because the genes for resistance were given to them by the Spanish when they came over. That's how nature behaves until something bizarre comes along. Oh, like, for instance, the West Nile virus. So we introduce a brand new virus into the new world from the old world, and all of the old world critters survive, and all of the new world critters don't. Simple story, right? It gets more complicated. But you all follow this so far, right? Okay. So we know now that the West Nile virus is just another member of the flavivirus family. You know what flavy means, right? All you Greek scholars? It's the Greek word for yellow. Right. So yellow. Yellow fever. I guess because it causes jaundice. So it's transmitted by a mosquito. A very famous mosquito called the yellow fever mosquito. So they've named it after the virus. St. Louis encephalitis is a mosquito-borne infection. Western equine and eastern equine encephalitis are vector-borne, mosquito-borne infections. Lacrosse virus. Anybody here from Wisconsin? Because that's where lacrosse is. Dengue fever. That's something else you have to watch out for when you travel. These are all vector-borne diseases. So the flavivirus group is, in general, or in specific, I should say, vector-borne. They're mosquito-borne infections. Now, what you have to know about is, well, okay, but how many different kinds of mosquitoes are there? And how many different kinds of hosts for each one of these are there? And the answer is, this is a highly restricted virus. It infects primates and humans. Well, we're primates. Non-human primates and humans. This infects primates, okay, but it's really bird parasite. And of course, named after St. Louis, because that's the epicenter for where it was discovered. And eastern equine, you can guess at the host species here, right? It's horses, sure. And it's an awful disease because if human beings catch this by accident, and it does happen, it's almost uniformly fatal. You die from this. So people are really frightened of this one. But there are seasons for these things. Lacrosse virus, not so bad in people. Mainly a bird parasite. And dengue fever, only in people. Only in people. But all of these are carried by mosquitoes, including the West Nile virus. So if there's the flavivirus group, then there must be something specific to the group, which you can say, all of those viruses have at least one thing in common. Well, for instance, they're all about the same size. So that's too bad because you can't tell which virus you're looking at simply by looking under the electron microscope. It won't tell you that. So God bless. Another thing that they have in common is that some of their proteins are so similar to each other that when you make an antibody against one, the antibody will detect all of the other members of the group. So we would call that a group-specific antigen. And that's exactly what happened in New York City in 1999 in the summer when the serum samples were sent to the Centers for Disease Control, and they looked for all of the virus infections that it could possibly be in the New World. They had a bank of sera that would detect all of these infectious agents, and they certainly did detect antibody levels against the St. Louis encephalitis virus, but it wasn't the St. Louis encephalitis virus. And you know who thought she knew why it wasn't? Tracy McNamara, because it was also killing birds. You can become Tracy for this time. So this bird here shouldn't be dying from St. Louis encephalitis. So either you have two different things going on at the same time, which seems unlikely, or you follow what they call Occam's razor. You know about Occam's razor? Okay. Used to shave every day with it. I don't know why, but he did. So Occam's razor says when you're confronted with two solutions to the same problem, pick the simple one first. If that one doesn't work, then go to the next. But try the simple one first. So the simplest explanation of what's going on is it's the same virus in birds and people. Well, how can that be? We don't have any viruses in this country that will kill birds and people at the same time. We don't have any like that. That's too bad we didn't think of that before we had to do all the experiments to prove what it was. So for a long time, we were chasing our tails, trying to find out more about this new variant of St. Louis encephalitis virus that gave a weak positive reaction with the antiserum that we had available to us at the time as a detection mechanism. It's called the West Nile virus because it comes from the West Nile district of Uganda. It was discovered in 1937 by some British physicians who thought that this particular patient had sleeping sickness, as I see some of you have today, particularly the gentleman in the back of the room. So it's okay, you can erase that part of the presentation. So sleeping sickness is caused by a protozoan infection, and it's transmitted by the tsetse fly. And you know this because of the New York Times crossword puzzles. Tsetse, T-S-E, T-S-E, fly. But it turned out that this woman was not suffering from sleeping sickness. She was suffering from a new disease that had never been discovered before. And so they gathered her serum up, and they saved it. And this was in just the very beginning of being able to detect virus infections by other means rather than just serum samples. we could look under an electron microscope and actually see what was going on. And the virus then was worked on by people living in Israel because they had a similar cross-reactivity with the same serum. So the Israelis began working on this in 1952. Israel had only been a country for a few years, right? And here they were already concerned about an infection. What's up with them? I mean, look at where they live and look at all the strife that they have to put up with that has nothing to do with a virus infection, and yet they concentrated a huge amount of their resources to this particular virus infection. You know why? Because they got it every year. Every single year. There was an outbreak of West Nile virus in Israel, including, of course, 1999. Uh-huh. It gets interesting. So what we know today about this infection, and these are not well projected, but trust me, these are absolutely gorgeous. What? I don't know. At higher risk of putting everybody to sleep? What do you think? I can still see the whites of your eyes, so don't you dare go to sleep on me. So West Nile virus is transmitted by mosquitoes. There are two kinds of mosquitoes. We just learned that today in our TWIP lecture. There are culicine mosquitoes and there are anopheline mosquitoes. Culicine mosquitoes transmit encephalitis, filariasis, all kinds of other viral infections, but they don't transmit malaria. Those are done by the anopheline mosquitoes. So mosquito vectors are just two kinds, and in this case, it's a mosquito species that would, if given the choice, always choose birds. Here's a human, here's a bird. Let's make the bird the steak. Let's make the human the hot dog. Well, if you're hungry and there's only hot dogs, okay, I'll eat a hot dog, but I don't like it. And we'll get back to that, because there is the hot dog theory of how this spreads to humans from birds. So ordinarily, it's an infection that circulates among birds of different species and the mosquitoes. Now, if the West Nile virus comes from the West Nile district of Uganda, and the mosquito that spreads it is called Culex pipiens, where do you suppose that mosquito came from? Don't you love the way I ask questions? Don't you wish I was giving you your final exam? Because they're so easy. I want you to learn this, so I don't want to trick you. I want you to remember this. So if the virus that's carried by this mosquito comes from the West Nile district of Uganda, where do you suppose the mosquito that carries it comes from? The West Nile district of Uganda. But there's a catch. The catch is that the mosquito got here first and had to wait, how long? A long time for the virus to find it. And that's what happened in this country. The virus found its vector in the New World. So how do I know that this mosquito species comes from Uganda or Central Africa? Well, by genetics. It's an easy thing to do, actually. So Culex pipiens complex. There's a bunch of mosquito species with similar names. They're sibling species. They look alike, but they've got different genetics. All come from the Central African region. And this virus is an African virus. And that explains why the birds from Africa didn't die at the zoo. Because they'd already seen it. Their immune systems were going to die. Oh, that's just West Nile. So what? Oh, that poor flamingo. Did you hear about that poor flamingo? Yeah, and the Himalayan duck. Oh, you should see that duck. It's just... So when this virus arrived on our shores in 1999, and I'll get to how I think it got here, the mosquito vector was already here waiting for the virus. Now, where do you suppose the virus came from? And at what part of the world do you think it arrived at? You have all the clues now. Come on, I know you know the answer. Say it. Oh, that's a great guess, though. That was a great guess. It's a great guess. But no. Yes? No, that's the Asian tiger mosquito. That's another good guess, though, because that mosquito lays its eggs on the rim of the tire, and then the water has to go above that in order to hatch the eggs again. That's why they can survive from one place to the other. They came from Japan, by the way. No, what are people doing at the Bronx Zoo? They're creaming these mosquitoes that are biting the heck out of them, right? All kinds of people are at the zoo online waiting to see the gorillas, including some little kids from... So I love watching light bulbs go on. I taught for 38 years at the medical school, and I must tell you that 99.9% of those light bulbs were already on. I won't say which ones weren't, but we all knew which ones weren't. But it's nice to see light bulbs go on for the first time. So that was a light bulb that went on just now. So my hypothesis, and others too, involve an outbreak of West Nile virus in Israel during that summer, and it was well documented. You know, that's just speculation, though. What are you picking on poor Israel for? Come on. I'll tell you why as we develop the story. But what we know about this virus, based on prior knowledge, was that it's a bird-mosquito cycle. And in Africa, no birds die. But when these birds migrate out of Africa, on their way to the Middle East, for instance, and on their way to Russia when they mate and lay their eggs and raise their young and come back to Africa, they take their virus and they bring it to those parts of the world, and they spread it to local mosquitoes that give it to local birds that didn't grow up with it, and they kill them. So one of the things we learned about this outbreak in Israel was that Israel was raising tremendous numbers of gray geese for France as a source of foie gras. And the geese in Israel were absolutely, they weren't decimated. You know what that means, right? Just 1 in 10. This was like every goose. Millions of geese were dying from West Nile virus during the migration of the birds out of Africa into the Middle East. When little kids catch this virus in Africa and other places too, they can get sick or not, but they can carry very high loads of virus. So high that the mosquitoes, if they bite those little kids, can actually pick up the virus. Now that doesn't happen in adults. In adults, we get sick right away. They take us to a hospital, and there's probably no chance at all of mosquitoes biting us and then transmitting the infection to others, because the virus titers in our blood don't get high enough. Now, what do you think the difference is between little kids and adults? Well, one of the big differences is the fact that your skull has not yet fused with your sawtooth joints. And as the result, when you get an infection in your brain and it starts to swell up, the joints just come apart and your brain sort of just makes it bigger. But you don't get sick because the encephalitis that you get doesn't press upon the brain case. In an adult that's hard-headed, most adults are hard-headed, the brain swells up and there's nowhere for it to go and you get sick just like that. So little kids can harbor higher levels of virus without letting on that they're sick. So our hypothesis, and I say our because there's a lot of other people that believe this is true too, during this epidemic in the geese, some little kids living on kibbutzes also got infected and got invited to come to visit their relatives in Queens and said, "We're going to take you to the Bronx Zoo to see 19 lowland gorillas. How do you like that?" Who would say no to that? I wouldn't. So away they went, and on line they stood. And that's how we think the West Nile virus came to the New World. Now, there are other ways of proposing this, too. You could say a stray bird got blown off course. Unlikely. A mosquito snuck on board an airplane and then snuck back off and then gave it to somebody. That does happen with malaria occasionally. It's called airport malaria. But the likeliest place for this to come from is some infected individual. A little kid. I checked out the number of people that fly every month from Israel to the United States and back in the summer. It's around 300,000 people a month by El Al Airlines. That's just one airline. And how many people then does that have to take in order to jumpstart this infection? But there are other things that work here, not just the people and not just the infection. You need the right weather conditions too, as we'll see. So we're an incidental host. We are an accident. We have no real role to play in spreading this infection. Other animals too, these are horses over here. And in fact, if you're a horse, this infection has a 40% mortality rate in horses, showing you that this is not the real host for this virus. That's one of the big reasons we want a vaccine for it. Because if we don't have a vaccine for West Nile virus, then the European horse racing industry will not bring their horses to this country to race, because they do not want them to encounter West Nile virus. And we know who controls the racehorse industry in this country, don't we? But that's another show. I see you have an FBI shirt on. The FBI would know the answer to that, of course. It's in wildlife. Wildlife, 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 wildlife. How many different species of birds have we now found infected with West Nile virus since 1999? And the answer is over 130 different species of birds harbor the West Nile virus, and many of them die. Robins, blue jays, and crows in particular. The corvidae. So what are the conditions necessary for spreading this infection, not to birds? We're not interested in birds. We're interested in people. Forget the birds, because birds will always have it now. But this infection, established in 1999, it'll never go away. It's always going to be here. It's going to be here. Here's an infection that you can say, in my lifetime, this was introduced into an enlightened country with an intact public health system, and we could do nothing to prevent its spread. These are the conditions under which this virus stops being found in birds and starts being found in people. What is the difference? I'll explain. Of course, that's why I'm here. Or at least I'll give you my explanation. So one of the explanations is that, well, if it's cool and wet, then you'll always see birds. And you'll always have mosquitoes, like today's rain. You have another day like today with rain, that water will stay around for a week and a half, 2 weeks. That's enough for mosquitoes to reproduce in it. And you will have a population of mosquitoes that you didn't have a week and a half ago. Think about that during your next visit to the zoo, because it's something to contemplate. But if you're a Culex pipiens mosquito, you prefer steak to hot dogs. And as long as there's steak on the plate, they will prefer to eat the steak. Now, how do you keep the steak on the plate? How do you make a bird happy? And the answer is, give them plenty of water to drink, give them plenty of food to eat, and they're happy. So what is that to a bird? Well, in this case, to crows and to other birds, it's insects, pollen, lizards, who knows what birds actually all eat. All those different birds eat different things, seeds. All these things are around when the weather supplies the environment for that. The water, of course, allows the mosquitoes to breed. All of them. Take a guess how many different species of mosquitoes there are in the New York City limits. I'll give you some choices. How many think 10 different species of mosquitoes? Just raise your hands. How many think, this is not a trick question, 20 different species of mosquitoes? Do you know this answer already? Okay, good. 30 different species of mosquitoes. Still no hands? This is no fair cheating. You're not allowed to look around to see who didn't vote. And oh, if they're not voting, I'm not going to vote. 40 different species of mosquitoes. Maybe I'll emphasize this with my voice. You can guess which one is correct. 63 different species of mosquitoes. Oh, you think it's 63, do you? Yeah, then you'd be right on that one. We have 63 different species of mosquitoes, but only one of them transmits this infection both to birds and to humans. Only one, when you take away the steak, will eat the hot dog. The other 62 species either don't prefer humans at all, so they don't bite us, so we don't even have to worry about them, or they don't prefer birds, so there's no way for them to pick up the virus to begin with. So we have one mosquito species, one out of 63, that's a prime vector for this infection in New York City. And it's the only species of mosquito that prefers polluted water. It's the only, there's a species of mosquito that prefers this. If you go to the 145th Street Water Pollution Control Center and talk to any of the maintenance people there, they will tell you legendary stories of how many larvae of mosquitoes, before they develop to adults, are present in the last batch of wastewater before it's dumped into the Hudson River, which is salt water at this point, so therefore it would kill those mosquito larvae. They're so thick that it's like a slurry of mosquito larvae. It's not a thin, watery solution of that. It's a slurry of mosquito larvae. And it's of one single species. Now, there are 14 treatment plants in New York City. 14. Are there other sources of polluted water in New York? Do bears? Well, you know the rest. Of course they do. So these are peridomestic. Peridomestic means it loves living near people. Why does it love living near us so well? Because we are so good at polluting water. We're the champions of the world. I'm from Louisiana. What's happening in Louisiana right now? Well, you'll find out. You'll just listen to NPR tonight. You'll see it. It's all on the news. It's the oil slick that we've created off the coast, right? It's going to probably trash all of Europe also, unless we do something about it. That's pollution. We know how to do that. Come on. Then we can clean it up, and you can create two industries. What's wrong with that? You know. So this mosquito likes to live in polluted water. So name some other sources of polluted water. How come this thing broke out in northern Queens, South Bronx? What's going on? Remember I told you about the Egyptian goose pond, or they called it a pond, but it was really another slurry of yucco, which, by the way, once they found out it was a vector-borne infection and they thought it was St. Louis encephalitis, they drained the pond. And people stopped doing this. I was there in September, dry as a bone. I waited online for just 2 hours to see the gorillas. No mosquitoes. How about Woodlawn Cemetery? Little cups that hold flowers for your loved ones. And you go to pray or say a few kind words, and at the same time, you'll be joining them soon. So it's difficult to prevent polluted water. Now, the unusual aspect of this outbreak in New York City in 1999, it required, and I hate using this expression, so I will use it, the perfect storm for all of the conditions necessary to bring the West Nile virus from the old world to the new world, environmental conditions to drive away the birds and make these mosquitoes choose the hot dog rather than the steak, and polluted water like you wouldn't believe. In fact, they didn't believe it. What are you talking about? Polluted water in northern Queens? Come on, that's an affluent community. Well, it happened to be the hottest, driest summer on record in New York. 1999 was the hottest, driest summer on record. We may break that this year, but we didn't break it for the last 11 years. What did that do? Well, you know what it did? It drove up the price of electricity. Huh? Yeah, that's what Con Ed does, because why? Because you have to use it. Why? Because of air conditioning. Oh. So the price of electricity goes up when the usage is in demand. Does that make any sense? Sure, if you're Con Ed. That's why they call it Con Ed. It's true. I mean, it was ridiculous. And so you know what happened? The people with in-ground swimming pools that had pumps that kept them from becoming polluted turned off their pumps and went on vacation somewhere else and said, see ya. We're going to be back when this heat wave breaks. We can afford to go away. In the meantime, their swimming pools became breeding grounds for these mosquitoes. And people began to have lawn parties that couldn't afford those swimming pools but lived in the adjacent neighborhoods. But the party wouldn't start until 10 o'clock at night when it started to cool down. When do you suppose the Culex pipiens mosquito begins its feeding frenzy? Around 10:30. How come that's true? Because that's when the birds go up in the trees and start to sleep. And when birds sleep, you know what they do? Well, they breathe in and out, just like you and I. So when you breathe in and out, what happens? You breathe in, the oxygen level goes down a little bit. When you breathe out, the carbon dioxide level goes up a little bit. You breathe in, the oxygen, all right, so you got the idea. It's a simple point to make. But if CO2 is the attractor for the mosquitoes, then how perfect is that? A bunch of crows go up in a tree, they all settle down for the night, they start to snore, down goes their CO2 along the trunk of the tree, out into the grass, out into the field, and the mosquitoes go, okay, here comes our meal. And they follow the trail of CO2 until it gets more and more concentrated. They fly up the tree, and they bite those birds. And that's where they get their blood meals from. How do I know that? Do you think I just make that up? Well, I'm capable of it, but I didn't. Someone at the university that I got my PhD from, the University of Notre Dame, actually sent graduate students into the field to measure the CO2 concentrations from rooking crows and the concentration of CO2 necessary to mobilize the Culex pipiens in the grass. That's cool stuff. For the major professor who gets to go home and have a nice meal and go to sleep, and the students get to spend all night measuring migration habits of Culex pipiens. Why? Because they also carry lacrosse virus. So he was interested in lacrosse virus. So he had them look at the crows, the CO2, and the mosquitoes. I must tell you that I actually published a little book on this subject called The West Nile Story, and you'll never guess who my two science advisors were for that book. Well, they're both in this room. And the only reason this book is actually truthful is because they forced me into it. Otherwise, I would tell you lots of these kinds of stories. So okay, so here you had the initiation of an epidemic. 7 cases. 5 deaths. Tragic. Meanwhile, they're working, I wouldn't say feverishly, but that would be a very bad use of that word in this case, to try in all they could to find out what was causing this infection. So Tracy McNamara, Tracy, being the clever person that she was, sent all the dead birds out to Iowa. That's what I'd do. Get them out of here. I don't care where you put them, but put them out of here. But Iowa's a good place. Nobody lives there. Put them out of there. No. It's the National Veterinary Research Laboratory facilities at Ames, Iowa. And that's where they do the pathology on these birds. So what they did there was they took some of the bird tissue. It didn't matter where. It was a hemorrhagic fever. It killed the birds by just absolutely decimating their entire circulatory system. So 1 in 10 vessels would be killed off. The bird would bleed to death. They didn't get an encephalitis like you and I got. So the virus was neurotropic in people and homeotropic. It infected all the tissues of a bird. Okay, so they took a tissue and what did they do with the tissue? You guys are virologists now. You're not just students anymore. You're at the door of your careers as virologists. So you take this tissue and you make a soluble extract from it. And you make a clear supernatant by centrifugation. But you don't centrifuge it enough to remove the virus from the clear supernatant because the particles are really small, right? And you inoculate that into other birds? Sure. Chickens, killed them. Ducks, killed them. Wait a minute. I've got a St. Louis encephalitis virus here. It doesn't kill any of those animals. It can't be encephalitis. It can't be St. Louis encephalitis, you know. It can't be. But CDC said it was. And CDC knows best. CDC was wrong. They weren't about to admit that they were wrong. What happened was they grew up the virus in a tissue culture situation. And then they looked at, and it was plaques. They got plaques on these things. Come on, they put holes in there. Right? And so they could tell you how many viral particles were present in the dead bird tissue by looking at how many plaques that they could generate from a mill of supernatant. Okay, so you've learned all of that already. Then they grew the virus up, and then they grew it up enough so they could make an electron micrograph of it. And they said, aha, it's a flavivirus. And someone else said, big deal. Which one? St. Louis encephalitis is a flavivirus. And you know it's not that one. So how can you find out? Meanwhile, there was this virologist in California who got in touch with the pathologist in New York State and said, could you please send us some of the brain tissue from the victims, and we will PCR out the entire viral genome for you. What would you have said to that? Come on, Tracy, I know what you're going to say. You're going to say you're full of... You know why? Because no one's ever done that before. So what makes you think you can do it? This guy was sharp, though. I mean, he was really sharp. He says, I can do this. Come on, coach, put me in. I can do it. Give me that brain tissue. I know I can do it. How much do you want? Okay, we'll spare that much. So they shipped it out to him. The man's name was Ian Lipkin. You know that name? You don't? He's a Columbia professor. What he did was he put his crew to work night and day using the polymerase chain reaction to generate the entire genomic sequence for this virus. The first little snippet that he got, he put it on the internet and says, does anybody recognize this sequence? I think the question mark's down over here. And the answer came back immediately from Paris. Paris? No, not the god Paris. This was Paris, France. Vincent Deubel. The name Deubel means mollybolt or something. Dowel. I think it means dowel. So Vincent Deubel sent back an email and said, I know exactly what it is. That's the West Nile virus. Where'd you get it? Send it over. I love him. He didn't say it exactly like that. He says, send it over. I like them very much. I have the virus in my refrigerator. I've got 23 different varieties. You want to see some? I'm not joking. He had 23 different varieties of them. How can you have 23 different varieties of the same virus? Well, you already know the answer, don't you? There are 23 different places in the world that they were isolated from, and each one gave a different sequence, slightly different. How slightly different? 90% the same, 10% different, 15% different, 23.2% different. So he said, when you get some more of this, let us know. Meanwhile, back in Iowa, the Iowans sent off an email to the Centers for Disease Control in Colorado. And in Colorado, there's a vector-borne disease laboratory as part of the Centers for Disease Control. And they said, please send us all your reagents for the flavivirus group. We would like to examine what we have in our serum samples and on our plates for the flavivirus that we think is causing this outbreak in New York. And the response came back immediate, and it said, no, you send us the virus, and we'll tell you what it is. And it was done in that spirit and that tone. So, Tracy, what would you have done? I can think of a few choice words for this one. They all start with the letter F. The first one is, forget about it. That was the polite way of telling them. There's another way, too. But they weren't in that position. They couldn't say that. They had to send their viral samples to Fort Collins to Dwayne Gubler, of all people. A man by the name of Dwayne Gubler, who as a young man was an extra in John Wayne movies, and later on became the world's expert in dengue fever, which is a flavivirus group. And so he said, send me your virus, and I will tell you what it is. Meanwhile, Ian Lipkin's group in California got more sequence and more sequence. Now, the virus that was sent to Fort Collins was then sequenced. And the virus sequenced from Iowa and from California was the same. It was identical. Cool. Except, of course, that the group at Fort Collins took full credit for it, not realizing that there was a group in California sequencing it night and day. Because by the third day, they had the complete sequence. Ian Lipkin's group. And they said, we know exactly which strain of virus this is. It's from an Egyptian goose in Israel. Really? It's one base pair different. I think that was in a sequencing error. The biggest other difference of strains that they had was like 10%. I forget how big the viral genome is, but it's pretty big. 10% of this genome is a lot of base pairs different. So Ian Lipkin, in 3 days, from the brain tissues of the victims, sequenced out the same virus genome as was in that poor South American flamingo and that poor Himalayan golden duck. I didn't tell you the full name, but now I can tell you it's golden duck. And this explained everything. Oh, no wonder the birds are dying here. They've never seen this virus before. And no wonder the people are dying, because they were misdiagnosed. And, gee, I wonder what we're going to do now. Wouldn't you like to know what happened since then? So it turned out that throughout the world, the West Nile virus outbreaks were always associated with droughts and high temperatures. Droughts and high temperatures, but I just said this is a mosquito-borne infection. How can that be? Mosquitoes need water. But it's a peridomestic mosquito. And it lives on polluted water. And we're good at that. So we have to have water. 2.3 liters every day for us. So you turn on your lawn to water it. You shouldn't do that, by the way. It's just turning on your lawn mower or lawn watering device. And then the faucet drips a little bit, and there's a little puddle underneath the faucet, always, because you didn't turn it off completely. Or you got a bird bath. Well, there are no birds. Because why? Because when it gets hot and dry, the birds go away. They can fly. After all, even the sick ones. Leaving behind, of course, those poor little mosquitoes to now have to deal with their hot dogs. Us. So hot, dry weather drives away the birds, shrinks up their food supply, and forces them to move to another place, leaving us in the lurch. And that's when the virus switches from mosquitoes and birds to mosquitoes and people. This has another very interesting ramification in that the virus replicates in mosquitoes vertically, meaning the eggs of the mosquitoes contain the virus, the larvae of the mosquito contains the virus, and the pupae of the mosquito contains the virus. And when they hatch, the adults contain the virus. You don't need to bite another victim to spread the virus from mosquito to mosquito. Now, when there's a big drought, like the one shown here, and then it rains like hell, why? Because that's the definition of a drought, isn't it? The drought starts with no rain, a long period, and then it rains. The only survivor of that drought is the pollution-loving mosquito. So it grows the population of mosquitoes that are left, namely Culex pipiens, so it gets even worse for the next 2 or 3 weeks. And here is a model of what's going on. Here's the weather. Here's how the temperature, when it rises, increases the viral replication rate in the mosquitoes because the air temperature is hotter. When temperature, you know this rule, right? For every chemical reaction replicate inside of a mosquito at a rate that's governed by that rule. So if it's low air temperatures, low virus replication, low numbers of viral particles, below the infection limit to infect people, how can that be? Is that really true? Can you actually get bitten by a mosquito, they inject virus, and you don't get infected? And the answer is yes. Absolutely. You can prove it in mice by injecting various low doses of the virus. And there are doses below which the immune system of the mouse is quick enough and soon enough to intercept that virus and kill it before it starts its replication cycle. Bird behavior is a big deal here too. Okay? So the birds are driven away by high temperatures and low precipitation, forcing the virus to move from mosquitoes and birds to mosquitoes and people. Now, let's see if that's true. How can we do the experiment? Well, that's easy. Nature already did it. It's already done. I wouldn't be standing here before you today if I... It's true. I mean, in 1999, remember I lost my grant. I'm depressed. I'm thinking of, I don't know, selling encyclopedias or something. Who knows what old professors do when they lose their grants. But I elected not to go that route. And so I glommed onto this. Barbara Nichols was great. She went out and collected all of the data. She went to the library. Remember those things where you used to keep books? She went to the library and she Xeroxed every single article ever written of the West Nile virus, which was a stack of reprints this high and this high. And then one day, in my air-conditioned office, I might say, we sat down on the floor together and we made piles of reprints based on the topics. Biology, virology, meteorology, sociology, geographic location, all kinds of articles published on this virus since 1937. And all of the ones dealing with the outbreaks, we then read thoroughly. And every single one of them had the comment in there, we don't understand the outbreaks with regards to weather. It occurred during hot, dry periods of time followed by an intense rainfall. The biggest outbreak ever recorded prior to the one in the United States was in South Africa. The hottest, driest summer on record. So here's the way it looked in 1999. These are the cases. Hottest, driest on record. Human cases in 2000. Still hasn't spread very far. 2000, 2001, 66, but notice now it's starting to spread down the Atlantic seaboard. West Virginia doesn't have a reporting service. They never did. They never will, so you'll never hear about it in West Virginia. But it's there. It's got to be there. I don't care who you are. In South Carolina and West Virginia, they sort of talk with each other, but that's what's going on. 2002. That's just how many years? 1999, 2000, 2001, 2002. 4 years of history with this virus. And we have it in almost every state in the Union. These are the states that were spared. And these are all the states that reported. And you talk about drought. You talk about high temperatures. This was a big, big year for that. 4,000 human cases. Let me tell you something else. They went back in the South Bronx and in northern Queens after the outbreak, and they drew a big circle around the epicenter where they thought it was, and they took random serum samples inside from people to find out how many people living inside this circle might have had antibodies against the West Nile virus. Now, they could go back in time to 1998, 1997, 1996. None of the serum samples in any serum banks anywhere in the world, anywhere in the northern hemisphere, anywhere in the North American hemisphere, had antibodies against the West Nile virus from people that had never gone to places where it was found. So we knew it wasn't here before 1999. When they went back and did this thorough survey, they found an estimated 8,000 people had encountered this virus. There was only 62 cases. Wait a minute. What are you trying to tell me here? Are you trying to tell me that, and most of those cases were older people, And all of the deaths were in older people. So people of my age group, 42. Okay. 52. Died from this infection. And you guys would have all been spared. Isn't that interesting? So you'd have, for every case that comes to the clinic, you got 200 people out there that also got infected. And not infected enough to actually have it reported. So here we have 4,156 cases. Who's got a calculator? Come on, somebody in this room's got to have a calculator. Multiply this number times 200 and tell me how many people were actually infected that year. That's a lot of people, right? You just add the zeros, what is it, 41,000 people? No, it's 81,000 people. It's a lot of people. What about the next year? Are you kidding me? It was even hotter and drier that year than it was the year before that. And now, everybody except Washington and Oregon, where it always rains anyway, so it's never going to happen there. Well, maybe. Almost every state in the union reported cases, including Texas, big time, and California. You watch these two states from now on. These are the places where it's endemic and it'll never go away, and you'll get a lot of sickness and a lot of deaths from this virus. Look at how complete this is. Now multiply that one times 200, and you get a big, big number. That's how many people were infected. We've had this infection so long in this country that people are now getting it by organ transplant. You go in for a kidney transplant or a heart transplant or a liver transplant, and one of those victims that was killed in an automobile accident that you got the liver from, they had West Nile virus. Guess what? You died from it. After all of that expense and all of that agony and all that suffering, to die from an infection that we never even had since before 1999. 2005, 3,000 cases. 2006, 4,000 cases. 2007, 3,000. 2008. And then, gee, what happened here? I know. Do you? Yes, you do. You know the answer already. You do? I already told you the answer. Why? What would be your best guess? This is an exam question now. The biggest difference between 2009 and 2008, speculate. What's the big difference? Huh? Thank you. It was a cool, wet summer. Plenty of broids, plenty of mosquitoes, plenty of people. But who cared? The birds were having steak for dinner. And we were spared this infection. In 2005, in October, there was an outbreak of West Nile. This is in Russia someplace. It's down over here. And the peak of morbidity was recorded in September, which this year was an unusually hot and dry. And there was only one outbreak that I know of where the weather wasn't hot and dry, but it involved a lot of polluted water. So we could probably use that as an exception to the rule. But in no other case has there ever been an outbreak of West Nile virus in which it was not preceded by a long period of drought and hot. If it was droughty and cold, still didn't have people involved. If it was wet and hot, like today, you still wouldn't have people involved. It has to be hot and dry. That's actually your exam question from me. So, I've taken up a lot of your time. So, sorry that I took so long with this, but I think it's instructive that there are a couple of lessons here. Number one is that know when you've been lucky. What was my luck? Losing my grant. How does that figure into this? Not according to some who would have said losing your grant is probably the worst thing that could ever happen to you at this kind of a place because, well, how are you going to eat? Or they can say, well, what else can you do? So this has the greatness to it and the flexibility and the diversity to have allowed this to happen. So I am eternally grateful for that fact. Because otherwise, if I was at, let's say, my alma mater where I got my undergraduate degree at Fairleigh Dickinson University in New Jersey, they would have said, you know, it's really been nice knowing you. You know, we can't afford to keep you because we don't know where your money is going to come from. So now you know where my money came from. Thank you all. Barbara Nichols didn't want any part of the publication, although I offered her authorship on it, and she didn't take it. And so I authored the book myself. And it's written in a jocular way that you can imagine, but it's got serious underpinnings. And so I haven't updated this book. I wrote the book in 2000, and it's a tongue-in-cheek romp through an outbreak of a virus infection, which has little or no serious consequences unless, of course, you die from it, which has a very, very low mortality rate now. Why? Because we know how to recognize it. So what's the first thing you do when you have a victim of West Nile virus? What's the very first thing you do as a physician? They're suffering from malaise, they have muscle weakness, and they're confused. Sounds like somebody like myself, for instance. Here's what you do. You check their antibodies, and at the same time you start them on steroids. Steroids. Now, no, not those steroids. You know, Arnold steroids. Those are other kind of steroids. These steroids inhibit the immune system, but it also keeps down swelling. It allows your brain to suffer through the viral infection without the consequences of the swelling part. And so the mortality rate for this infection has gone from around 6 to 8% down to less than 2%, except in Texas. Anybody here from Texas? Yay. In Texas, they have unusual health insurance regulations. Even more unusual than the rest of the country. They depend a lot on migrant workers. None of the migrant workers are protected, whereas in California, all of the migrant workers are protected by health insurance. So if you get sick, you get cared for. In Texas, they try to send you back, and you might actually make it or not. So the mortality rate in Texas from West Nile is twice that of California. And unless they change their laws, it'll always be that way. Which lets you look at sociological consequences of infections also, rather than just the biology of them. There's a big consequence to pay for this. And that is, what if you were living in, let's say, Scottsdale, Arizona, which most of us will never be able to afford to do, but let's say we could. You know what the average age of a citizen of Scottsdale, California is? About 67. That's like a monoculture of death for West Nile virus. That's anybody who gets sick from West Nile virus in Scottsdale has a good chance of dying. Oh, but they have pretty good health insurance. In fact, they have incredible health insurance. They get cared for no matter what. A hangnail, they've got a special clause in one of their policies just for that. So those people will never die from this infection. And it's sad because there are lots of people out there that are dying from this, not just in this country, but in other places too. It's an infection that you don't have to die from if you can catch it early enough. And it's a lesson in how diseases spread and the conditions under which they need to spread. Interesting side note. I tried like hell to obtain the specific data for outbreaks per state, per county, per block of city. I just wanted to know their zip code. And I wanted to know the exact time of admission into the hospital so that I could do the following. Let's say Illinois. Here's the state of Illinois. And here's June, let's say May, June, July, August, September, November. And here are all the counties. Now, just in your mind, just close your eyes and imagine a black dot appearing in, let's say, mid-June, early July in DeKalb. And then 2 more dots in Cook County, and 10 more dots in Urbana, and 200 more dots in Cairo, and then 2,000 dots in, let's say, the entire state, and then then this timeline is moving along, and then in September it starts to go down, and in October it's over. And I want you to see that movie, because that movie tells you how these diseases move through populations. And I wanted to make that movie for the entire country. And I wanted to do it for the drought years and for the non-drought years. How does this virus move from New York City to California in 3 years? Come on, Tracy, I know you know the answer. It's crows are the victims.
Right, and then crows fly everywhere.
They do, but they can only fly for 2 days when they get sick. Then they sit for a day and then they die. So crows are found. Do you drive at all?
Not really.
How about being driven? Sure. So you're driving down the highway with your parents or with your boyfriend or with whoever you're with, and you're driving along, like, say, Route 80, and Route 80 is a big ditch in the middle. There's on-side traffic over here, you're going this way, they're going that way. This ditch right now is filled with water, guaranteed. That water is a breeding site for mosquitoes. Who feeds along the roadside? Crows. Why? What do they find so exciting about the roadside? Do you know the answer? No. Say again? Roadkill. Which roadkill? What roadkill? No, they're eating dead insects that your windshield is killing for them. And they know that. It's creating huge bonanzas of insects. And so the crows are all the way along the side of the road feeding. And right over here, unbeknownst to them, are breeding populations of Culex pipiens. So when your car goes down the road at very high speed, 70, 75 miles an hour, the draft that your car creates drags the mosquitoes to the next spot. Now, I proposed this in 2000 without much data. It shows you what a small world it is. I actually sent a letter to Time magazine telling them that's how I thought this virus is spreading from the East Coast to the West Coast. This was in like 2002 or 2003. And a friend of mine that I haven't heard from in 25 years wrote me a letter and said, "Is this the Dick DePalmier I grew up with in Dumont, New Jersey?" And I said, "Yeah, that's me." He says, "I read your letter in the Time magazine." So how would you prove that? That the drafts of cars actually moved mosquitoes from place to place. And then new batches of crows got infected. And then they brought it to some little place. If you could make a movie of the outbreaks, you could get that data, right? Just like that. So I tried to get the data. I'm almost done. The states wouldn't give it to me. They told me to go to the CDC. The CDC wouldn't give it to me. They told me to go to the states. Do you know why neither one would give it to me? Because of homeland security issues. This is not just an ordinary virus infection. What if this was a really dangerous virus that was also mosquito-borne? I would now know exactly what to do, when to do it, and how to do it. You want to know what's next? Stay tuned. Thank you.